Okay, so today's video is going to be about cell cycle and mitosis. So let's first just kind of take a look at the cell cycle. So this whole thing here is the cell cycle, and the cell cycle is divided basically into two parts. Right? You've got interphase here, okay, which makes up the majority of the cell cycle, and the interphase has three different phases that the cell can be in. What's called G1, S, or G2, and then you have this other portion of the cell cycle here, mitosis. So the cell is going to spend the majority of its time in interphase. It is not going to be dividing most of the time. Cells do need to reproduce or they will die. Okay, and so the cell cycle is going to determine the cell life. It's where it is in the cycle will determine whether it's going to reproduce or not. Cells that do not reproduce are what are called terminal cells, and so they kind of freeze in the cycle. So let's take a look at what that is. So what we have here is what is called G naught. Okay, and so that's this G0 here. This is G naught. So we have interphase, and so all of this portion here is interphase, and this side here is mitosis. And so you've got some cells that never make it out of this first portion of interphase, this G1 phase of interphase. And so these cells that are not reproducing, or cannot reproduce, are what are called terminal cells. And so terminal cells are stuck in this phase here that's called G naught, or G0. And so these terminal cells cannot reproduce, and so they will eventually die. They will not be able to replace themselves. And so these are things like red blood cells, nerve cells in our bodies, some muscle cells. And so if they die, so they are not constantly regenerating, replacing, like say skin cells would be. So those cells are stuck in that G naught phase. They are not continuing forward through the cell cycle. Okay, so let's focus on what's called the G1 phase of interphase. So we're right here. Cell has recently come out of mitosis. Remember, this whole section here is interphase. So we're in the G1 phase of interphase. And so this is a cell that's going to go divide. Okay, it is not in that G naught phase where it's not going to be dividing. So during this G1 phase, this cell, called our gap 1 phase. Some books write growth phase, but your AP test will probably call it gap 1. So during this G1 phase, the cell is the organelles are reproducing. Okay, so organelles are being reproduced, more mitochondria are being made, more endoplasmic reticulum is being made. Sorry, I'm blanking on organelles, vesicles, vacuoles, Golgi apparatus. Organelle reproduction is happening. Okay, protein synthesis is happening. And for protein synthesis to happen, remember the cell has got to be able to access the DNA. So in this case, in this portion of the cell cycle, the DNA is going to be what's called loose or unwound, which is going to be in the form of chromatin. Okay, so that there's easy access to the DNA, to the nitrogen bases to be able to do transcription. This gap 1 phase, it can last for a really long time. It can be very, very short, or it can last for years. Okay, there are checkpoints during this phase. We'll talk about checkpoints a little bit more later, but there are checkpoints during this phase that will determine if it's going to be able to move on to the next step. Okay, and like I said, this is also where cells can enter that G naught phase. And we do have some of cells that are able to enter that G naught phase, and it can be reversible. They can get the proper signals from this proper stimuli, and they can revert back to that G1 phase. And so then they would be able to go on and divide. So then after the G1 phase, the cell will enter the S portion of interphase. Okay, and so the S portion of interphase is synthesis. Okay, and in this case here, we're synthesizing DNA. Okay, so this is where DNA replication will occur. So in humans, we'll take those 46 chromosomes and we'll turn it into 92. So the DNA will need to uncoil so that it can be replicated easily. The organelles will continue to replicate, and we have to have this phase to ensure that the new cells have the right amount of DNA. Right? If we didn't replicate the DNA, there'd be a cell with 46 chromosomes, and it would split, and they'd have 23. And that's not genetically identical. And when the cell is going through the cell cycle and then going to go through mitosis, it wants cells that are genetically identical to the parent. So we need to replicate this. So now we have 92, and when that splits, I get 46, 46. And now they're identical to the parent cell. So after that synthesis phase, there's another G phase or gap phase. So this is the gap 2. So again, we're still in interphase. Okay? G1, S, G2, they're all part of interphase. So in this gap 2 phase, the cell is really getting ready for mitosis now. It's making the proteins that it would need to be able to do mitosis, the tubules that make the spindle fibers. It's continuing to add organelles. It is getting organized, basically, to be able to then enter the M phase or mitosis. So as the cell leaves interphase, so it leaves this portion here. Remember, all this portion here is interphase. It will then enter mitosis. And your strict definition of mitosis, this is nucleus division. Okay, so I divide and replicate the nucleus, and then there's also a portion of it called cytokinesis, which will divide out the cytoplasm. But technically, mitosis is the division of the nucleus. That's why, like a bacterial cell, a prokaryotic cell, cannot do mitosis because it doesn't have a nucleus to divide. And there's many signals that will trigger all the different phases of mitosis that will help the cell move from phase to phase and interphase. This is regulated very closely with a number of checkpoints, and we're going to look at those in just a second. Okay, so let's take a look at this graph real quick before we move on. So this graph gives us the amount of DNA, and it's basically have two options. It's either a single set or duplicated DNA. And so it kind of traces out the amount of DNA that's occurring throughout the length of the cell cycle. So in this G1 phase, remember all of this here is interphase. And remember in the G1 phase, the cell is basically just going along doing its everyday thing. It's it's growing, it's making more organelles, okay? It's doing whatever function it needs to do in the multicellular organism, okay, in this first gap phase. But this is just a normal cell. And remember that synthesis phase, okay, this is where you're seeing this big increase in DNA, so that we get our duplication of the DNA. And remember the S was for synthesis, and in this case we are synthesizing new DNA. So in the G2 phase there, in that second gap phase, there is still twice as much DNA as should normally be in the cell because the cell hasn't divided and split that DNA out yet. Okay, the cell is now kind of finishing up, preparing to do that. It's setting up the tubulin and the proteins to be able to move things around. And so as it goes through mitosis, it's going to go through that division. And so when it gets down here near the end of mitosis, those chromosomes are actually going to physically be separated. And so now you're going to reduce your DNA in your two new cells down to the original amount that you had before. Okay, so far what we've been talking about has been mostly review. And when we get to mitosis, it should be mostly review as well. But in this control part now, if this is where you're going to hit most of the stuff that's going to be new, that you did not see your freshman year, you have not heard before. So let's talk a little bit about what controls the cell cycle, how it's going to get from phase to phase, and what is going to regulate the cell and its ability to divide. Okay, so most of the control of the cell cycle is going to be dependent upon what are called signal molecules. And it's going to be based upon the concentrations of these signal molecules. How much of them are there? As a general rule, there needs to be a certain amount, a certain accumulation of signal molecules to actually push the cell forward to division. Okay, if we look down here, most of these chemical signals, they actually need to be phosphorylated to be working. They may be present in the cell, but they're may be present in an inactive form. So for instance, these are my chemical signals there. I've got S and M. Okay, and until they are phosphorylated, remember, we've seen this term before. So until they are phosphorylated or they've had a phosphate added to them, okay, they are inactive. And once they get that phosphate added to them, then they can become active and they can trigger the cell through its division processes. Okay, so most of these molecules these signal molecules, they're called cyclones. Okay, you can see a variety of cyclones on this graph here. And hopefully what you'll notice is that their concentrations are going to vary throughout the cell cycle. And like I said before, you have to have a certain accumulation of these signal molecules for them to do anything. So you can see here is the accumulation of cyclone E. Here in the G2 phase, when there's a high accumulation of cyclone A, and then it will activate at the cell. Okay, and it will trigger the cell to progress through the cycle. And so most of these cyclones, what they do is they activate what are called cyclone-dependent kinases, usually abbreviated as CDKs. Okay, so they will activate these cyclone-dependent kinases. So these cyclone-dependent kinases are always, as a general rule, they're always present in the cell. They're just present in their inactive form. And so when the cyclone accumulates and binds to the cyclone-dependent kinase, and it will activate the kinase. And so once there's enough of that CDK activated, then that's going to signal the cell to move forward. Okay, so let's look a little bit closer then at exactly how a cyclone and a CDK work together and what a kinase really is. Okay, so as you can see here on the left, I've got these CDKs, these cyclone-dependent kinases. And they are, like I said, they're always present in the cell. And a kinase in general is a type of enzyme. Remember, ACE is enzyme. Okay, so a kinase is going to be a type of enzyme that's basically going to be able to do phosphorylation. Okay, it's going to be able to take a phosphate and it's going to be able to transfer it from a high energy donor like ATP to a specific substrate. Okay, so kinases are going to be able to do phosphorylation. They're going to be able to take a phosphate from ADP, add it to substance A, okay, and now you get ADP. So kinases are going to be able to transfer phosphates and phosphorylate specific substrates. And so a protein kinase is going to do that specifically for a protein, okay, which also could be another enzyme. And so these CDKs, like I said, they're present in the cell, but they're inactive. But the cyclones is what will activate them. And you can see here that this S-cyclone is activating this S-form CDK, okay, and the M-cyclone here activating that CDK. So the cyclone is going to bind to the CDK. And so once the cyclone binds to the cyclone-dependent kinase and they form that complex, now it's active. Now this kinase is going to be able to phosphorylate the signal molecules. Okay, so you can see it phosphorylating the signal molecules. And when there's enough of those signal molecules phosphorylated, then the cell will move forward. And as the cell progresses forward, you'll see that the cyclones are what are actually degraded or broken down. So the CDK, like I said before, stays present in the cell all the time. It's the cyclone whose concentration is constantly changing, and so that will cause those fluctuations in concentration that we saw on that graph a little bit ago. Let me pull that up real quick again. So you can see here my cyclone concentrations are what are changing. So as the cyclone is degraded in the cell, its concentration is going to fall. Okay? And so the cell has already moved